transport actually works. We have two main types of cell transport. We have passive transport and then another type of transport called active transport. Just like you would say someone is passive if they sit on the couch all day eating potato chips and not using energy, passive transport does not require energy or does not require ATP. Passive transport moves things from high to low concentrations. As we can see here, we have a cell and it has a high concentration of, let's say, salt. And the environment has a very low concentration of salt. So in passive transport, we would move things from this high concentration to the low concentration. So we see after passive transport, the particles that were in the cell have moved to the outside of the cell. And now we have an equal concentration with the environment and the cell. Alright, now let's look at some examples of passive transport. Our first example is diffusion. Diffusion is the movement of particles from high to low concentrations. As we can see in this example, we have a cell that is highly concentrated with, let's say, salt or sugar. And then after diffusion, some of those particles have moved out of the cell from this high concentration to the low concentration. So we now have particles outside the cell. So we have our before and after. There is a special case of diffusion. Sometimes those particles need help to move out of the cell or into the cell. And when we need help, we have to use a protein in the cell membrane to help us move those particles. This is called facilitated diffusion. If you were in elementary school and had a teacher's aide, you might have called that teacher's aide or that teacher's assistant a facilitator. Facilitator means to help. So literally this means diffusion that uses a helper protein or a facilitator. So facilitated diffusion is when a protein helps move the particles across the cell membrane. Now our other example of passive transport doesn't move particles, but instead it moves water. And that is called osmosis. So osmosis is the movement of water from high to low concentration. We move our water from areas with a lot of water to areas with less water in osmosis. So this is called a U-tube experiment, and this is our before osmosis situation. So we have side A and side B on the U-tube. As you can see, side A and B have equal water levels, but side A is highly concentrated in salt, and side B has a low concentration of salt. Since B has a low concentration of salt, that means it has more water. See all that extra white space? That means it has more water. That extra water is going to move from side B over to side A. So since we're taking water from side B, we expect the water level on side B to drop, and we're moving water to side A, so we expect the water level on side A to rise. As we can see, after osmosis, we have a higher water level on side A and a lower water level on side B. And that has worked to make our concentrations now equal on both sides of the U-tube. Now let's talk about our other type of cell transport. Our first type was passive transport. Now we have active transport. Active transport, unlike passive transport, requires the use of energy or requires the use of ATP. Our active transport pumps things from low to high concentrations. So if we can see in this example, our cell has a high concentration of salt and the environment has a low concentration of salt. So active transport will move these particles that are in the low concentration and force them into the cell that's already highly concentrated. So after active transport, we have a cell that has a lot of particles, or even more particles than before, and the environment has a very, very low concentration of particles. Alright, now for some practice problems. And these practice problems all involve osmosis. So for our first practice problem, we have a U-tube experiment. We have side A and side B. The first thing you want to do in a U-tube experiment is decide which side has more water. So we see side A has a low concentration of salt, side B has a high concentration of salt. So you see all this extra space on side A, that's all the extra water that side A has. So that extra water from side A is going to move over to side B. As a result, the water level on side B will rise because it's gaining water, and the water level on side A will drop because it's losing water. Now we're only moving water in osmosis, not particles, so the amount of particles on both sides will be the same as before. But as we see, since we added extra water on side B, we now have an equal concentration on both sides of the U-tube. Let's do another example. This time, side A is highly concentrated with salt, and side B has a low concentration of salt. So we're going to move that extra water from side B over to side A. As a result, the water level on side B will drop, and the water level on side A will rise. Again, in osmosis, we're only moving water. Okay, so we have the same number of particles as we did before osmosis occurred. But now the concentrations are equal because we've moved water to balance out the concentrations. All right, now for some more practice problems. What will happen if osmosis occurs? We have a cell that is 10% salt, and the environment is 5% salt. So when you're solving these problems, you always want to draw them out. Our box will represent the environment, and the circle will represent the cell. So now you're going to draw dots to represent the particles or the salt. Okay, the cell has 10% salt, and the environment is 5% salt. Since 10 is a greater number than 5, we expect the cell to have more salt than the surrounding environment. Now it says, what will happen if osmosis occurs? Since osmosis is the movement of water, our water is going to move from the area with more water, see all that extra white space, to the area with less water. So water is going to go into the cell, so water will enter the cell. As a result, the cell will get bigger. Just like if you add water to a water balloon, the water balloon gets bigger. If you add water to a cell, the cell will get bigger as well. And we can call that swelling. The cell will swell. Now, if there's too much water that enters a cell, the cell might eventually burst. Just like if you add too much water to a water balloon, eventually the balloon will pop. Now for our second example. A cell is 10% salt and the environment is 20% salt. So again, let's draw out our example. The box is our environment and the circle is the cell. Our cell has 10% salt, environment has 20. 10 is less than 20, that means the cell has less salt than the surrounding environment. Now you have to decide which way water will move. Remember, water always moves from areas with more water, and we see there's more water inside the cell. See all that extra space? And there's less water outside the cell. So we move our water from where we have more water to where we have less water. So as a result, water will move out of the cell during osmosis, and just like if you took water out of the water balloon will shrink, the cell will shrink since water is leaving the cell. All right, our next example. Cell is 10% salt, environment is 10% salt. As we can see, we have 10 and 10. Those are equal numbers. Let's see what happens. But first, draw out your experiment. We have our environment and our cell, and they both have the same concentration. So they both have dots that are equally spaced apart. You see, neither has more water or less water than the other. As a result, water will move both into the cell and out of the cell. Since water is moving both into and out of the cell will not change in size. All right, just one more example. This example is a little bit trickier. It's a real-world example. We have a freshwater plant that is placed in a tank of salt water. What will happen to the cells of the plant if osmosis happens? So our keyword here is osmosis. We're going to be moving water. Now, if you have a plant that grows in fresh water, it won't have a lot of salt. So let's go ahead and draw out our experiment. Here is our tank, and our tank has salt water in it. And then we have our freshwater plant. So let's draw our freshwater plant. Now, we would expect a freshwater plant to have less salt, maybe a few particles, but less salt than the surrounding environment. We said that the environment is a tank of salt water. So the tank has a lot of salt particles. Now, the tank is highly concentrated in salt, which means it has less water than does the plant. The extra water in the plant cells will leave and enter the surrounding environment. As a result, all of the cells of the plant will lose water and the plant will shrivel.